Good evening. In this seminar, we will present to you how depression exacerbates the hepatic steatosis in mice by activating the hypothalamic pituitary adrenal axis. This job will be presented by Mariana Parra and by myself, Miguel Paniagua.
The hypothalamic pituitary adrenal axis is an endocrine system that is integrated by a feedback loop of hormones. It's composed of the CRH from the hypothalamus, the pituitary ACTH, and the cortisol from the adrenal gland. It is related to the stress response. It also regulates metabolic process and the immune system.
Hepatic steatosis refers to the accumulation of excess fat in the liver cells. This can then lead to severe complications, such as liver dysfunction and other diseases that are very dangerous and can pose a threat to us.
The aim with this study is analyze how depression exacerbates hepatic steatosis in C57BL6J mice by activating the hypothalamic pituitary adrenal axis.
One test that was used for this experiment is the PCR test. Basically, this test, what it does is it amplifies on specific DNA sequences. It does so by heating and cooling off, so even small strands of DNA can be detected in the sample. It was used for three reasons. One, it was for assessing gene expression. Second was for the measurement of inflammatory markers, such as cytokines. And third, it was used for the investigation of metabolic pathway. Another test that was used for this experiment is called the restraint experiments. As the name implies, it is based on physically restraining animals in a set period of time, inducing psychological stress. And as we can tell in this experiment, stress is really important because it plays like a huge crucial part on depression and hepatic steatosis. It was made for two main reasons, for behavioral and psychological responses, and also for evaluating gene expression, which was also the goal of PCR test.
La prueba LISA es un ensayo de inmunoabsorción ligado a enzima. Es un método utilizado para detectar cuantitativamente la interacción específica entre un antígeno y un anticuerpo en una muestra. Este anticuerpo suele estar unido a una enzima que al momento de reaccionar se vuelve fluorescente y permite hacer el análisis. Se puede usar para detectar desde anticuerpos, hormonas, enzimas, virus y bacterias. En el caso de nuestro estudio se usó para analizar la CRH y la ACTH. La prueba Western Blot se utiliza para analizar la presencia de una proteína en una muestra por medio de electroforesis en gel, que separa proteínas según su electronegatividad. Las proteínas analizadas en el estudio son CRH, ACTH, GR y GAPTH.
Okay, so now we move on to the second result, which is going to be all about cortisol. So cortisol is a hormone that is released on high stress situations. One of the tests that we used was the restraint experiment, that it basically restrains the subjects in a limited movement, so it induces psychological stress. As we can see here, the cortisol affects the accumulation of lipids in the liver. So looking closely in the A image, we can see very few to none lipid accumulation. It is a very clear image, and it shows a very healthy liver. In the D image, however, we can see a very high saturated liver with concentration of lipids. So this one has the most of cortisol. Okay, so now we move on to these results, and what this basically tells us is if these ones on the bottom reduce or increase the concentration of these ones. As we can tell with BDNF and GAPDH, the concentration stays basically the same with all of these, so it doesn't matter. But with GR, the concentration does varies. For example, with CRS plus C group, the concentration of GR is very minimal. Meanwhile, with CON group, the concentration is very, very strong. As you can see here, it is determined by the darker and the thickness of the marker.
The first figure shows two rows of protein bands. The top row represents GR protein, the bottom row represents GAPDH protein. The CON and CRS groups seem to have similar GR bands intensities. The C, D, A, H, E, D, and C plus CRS groups appears to have less intense bands compared to CON and CRS, suggesting lower GR protein expression in these groups. The GAP-DH bands appears relatively consistent across all groups, indicating equal protein loading for each sample. In the results section show Mason trichome staining images of liver tissues from different experimental groups. There was no significant difference in liver fibrosis among groups. Similar overall appearance between the control group E and the treatment groups F, G, H. The treatments didn't induce significant liver fibrosis despite other observed changes in liver lipid content and metabolism.
Okay, so now we got to the discussion part of the seminar. As we can see, Han Y et al. Said CRS contributes to the abnormal metabolism of lipids in rodents fed with HFD. We agree with that statement. Also, Risky G et al. Said MCD is commonly used to induce models of NASH but causes significant weight loss in mice. We also agree. And finally, with Nard G et al., a positive regulation of BDNF has been observed in animal models of depression. Then again, we agree.
Our conclusions are: first, molecular biology techniques allow to measure gene expression levels and identify specific molecular changes associated with depression and steatosis hepatic. Second, analyze potential biomarkers related to depression and liver disease, which can be identified through molecular biology approaches. Third, molecular biology allows us to investigate using biological assays how various diseases affect the body and induce alteration in different biological systems and factors.